I think today's talk, I'm seeing it as like, what can we do next with RNA 3D? So I've titled it like, how does it work? But that kind of breaks down into two categories. The first is just what is RNA structure? So I'm gonna kind of present how I think about RNA 3D structure and what its building blocks are. But the second is how do we study it? So as more of you are kind of looking at 3D structures, how do you evaluate which ones to look at, which ones to trust, kind of very high level, what are the different techniques and what makes a technique more or less trustworthy. So just to start, I kind of wanted to lay out kind of my perspective so you guys know where I come from. So I do mostly, I'd say, discovery work. So that means I have some sort of natural sequence, and then I want to solve the 3D structure. For me, this is done with cryo-EM or computational modeling. So that's kind of the direction I take it in. But then there's the design problem, which is what Eterna has classically been focused on. So you have a shape that you want, could be 2D. In this case, I pictured it as 3D. And then you're trying to design a sequence that would give us this shape, right? And Riju mentioned it briefly in his introduction, but these don't have to necessarily be separate, right? So if I want a shape, so like this lambda shape here, and I give you a bunch of sequences. So we have an overabundance of RNA sequences that we know. Literally, there's people sequencing just the ocean to get more RNA sequences. Could you pull out one of those sequences that actually makes that shape? So could you select kind of natural sequences that would fold up into a nice RNA shape? And that's kind of combining discovery and design principles. So that's one pathway to kind of that we've been thinking about recently. I'm going to start with key thoughts about kind of, I guess, RNA 3D. The first, and I think Slava did a nice little intro as well, and I think seeing the CGEM models, especially 7K00, which I love as well, Sarah, we can learn a lot about RNA biology and how it works if we go all the way to 3D, so you can see those extra interactions. But it's been historically very hard to get those 3D structures, so we don't have it for all of the RNAs we know of. The second is knowing accurate base pairing actually makes solving the 3D structure easier. So everything I do is not possible without first knowing the secondary structure pretty much. So that's just your base pairing. So the work of Eterna has really helped us also predict those base pairing patterns as well. There's a great source of 3D RNA structures, which Slava showed. So that's what we call the PDB. Currently, it's unknown how many natural RNAs have 3D folds. We're really curious about that. We are not done discovering new 3D RNA folds, so there's probably more shapes that we haven't yet solved. And then the last things, right, is this kind of presentation is about kind of evoking more thoughts or takeaways or next directions that Eterna could go in the 3D space. So I think your thoughts and takeaways are even more important than what I am thinking about. So I'm going to split this into first just going over the levels of RNA shape, so how I split it up, then 2 case studies, which will show us different things about 3D RNA shapes, and then just briefly, and this will be throughout the case studies as well, how do we actually solve these 3D structures? And where can you go to find them? So the levels of RNA shape. So I like putting analogies on things, so I'm going to start with an analogy. And I split it into 4 different levels of kind of structural biology. So we have a scenario here where we have this scene and we have a character, which we don't know what the character looks like yet. And we are asking the question whether the character can fit through this tube, right? So can the character go through this tube and make it to the other side? the first level, we're just given a list of building blocks. So this would, be, okay, as red hat, brown shoes, etc. With that information alone, we're not really able to ask our main question, but we are able to get an idea that it's some sort of dressed being. The second level is how parts are connected, right? So now we can see, okay, the shoes are on bottom, the hat on top. Again, we have more information and we can learn a lot more. Now it looks human-like, but we don't know if it will fit inside the tube. Then we have 3D shape, and that's kind of where we start to be able to ask the question, can he fit? And in how it's currently shaped, right, he's a little too tall to fit through. But that's because we've ignored the fourth level, which is the fact that these things can move. So he can lay down, put his hands down, and then make it through the tube. And there's another part of this, so that's movement, but there's also interactions. So what if we want to stop him from crossing? Well, we can just give him a bunch of coins. Now he can't put his hands down and he's kind of stuck on the other side. So that's the fourth level, is how it interacts with other things and moves. So to bring this to biology, in this game, we have a scene. In biology, this is a cell, right? So this is where most of our biology is happening, this super crowded environment. In the game, we have characters, and in the cell, we have macromolecules. So this is the ribosome here, but we also have RNAs pictured in the pink here. Those are mRNAs. We have a whole slew of different players. So biology is when these macromolecules interact in a cell. And we're curious about a couple questions that the shape of these molecules can help inform us. So how does it work? In the case of a lot of what I work on, which is a lot of viral structures, how do we stop it working? So that's like when we gave Mario the coins. And in the case of a lot of Eterna work, we're curious, how do we actually design this to either change function, make it better, enhance it, etc. So to move to RNA, let's see what these 4 levels are. The first is just a sequence, right? So that's the sequence of your 4 bases. Then you have how parts are connected, which is the base pairing. So that's where Eterna has historically been focused on. Then we have the 3D shape, so how it actually folds up in 3D space. And then we have how it may interact with other things in the cell. So my work is mostly focused on taking a sequence and asking what is its base pairing pattern and then what is its 3D shape. Eterna has been historically focused on asking, okay, I want this shape in 2D space. What sequence do I design to get that? And then perhaps Eterna 3D can kind of move even farther and ask, okay, what if I want this shape in 3D? How do I get a sequence that makes that? So to start with 3D structure, I'm going to start very simply with a simple stack here. And I'm sorry, these are not in Eterna colors. This is, I think, one of the slides I totally forgot to swap the colors, but I promise these are real base pairs. So we have a stack and then we just have a simple loop. What does this look in 3D? Well, it's no longer just a simple ladder. In 3D, RNA will make a helix. So you see kind of a spiral staircase. You still see the base pairs, and they're still stacking on top of each other. They just do so in a helical form. And just to look at how that fills the space, right? So this is just a space filling model where we can see it's a nice helix. So that's the base of 3D structure, is really like, all right, we have helices. So let's take that to case study 1, which is going to be this PDB2NBX, which is a viral RNA. And this part of the RNA is very important for translation. So without it, the virus doesn't make its viral proteins as well. And this is what it would look like if we were to design it in Eterna. So we have, a nice pair of stacks here, another one here, a third here, and then we have a little extra fourth over here. So we pretty much have, a four-way junction with three large stacks or in 3D helices. And this we can get from the fact that, we know bases like to pair, so this is the base pairing pattern. But let's start to think about what this may look in 3D. And the first thing is we know all these are helices, right? So we know we're gonna have, an orange, a yellow, and a blue helix, and then a little green loop over here. Great, we know that, but how do they actually fit together in 3D space? And the first major, I guess, energetic quality we have to think about is base stacking. So we know in stacks, right, bases like to stack. So for example, here we have base stacking, and that makes up our helix. Well, this is also true at junctions. So we see here, right, we have three big helices all coming together. Well, they like to stack between helices as well. So here we have one coming in, and this is not connected. So the RNA strand goes this way, and then on this side it goes this way. And you can see even at the junction where we don't have a continuous RNA, we still get base stacking. So it looks like a continuous helix, even though we have the strands going their opposite ways here. So we have this stacking between our helices at this junction, but we have two possibilities. So we have actually the orange and the yellow can stack, as I have up top here, but then the blue and the orange can also stack, as I have here. And to be honest, we're not at the point that we can really predict which one's going to happen. So someone had to solve the 3D structure for us to know which this is. And luckily in this paper, they did just that. And we knew that the orange stacks on the yellow and then the blue is separate from that. So this is what it looks like. That black image was actually the real structure from the PDB. And you can see the yellow here and then the orange here, and you can see a nice, pretty much looks like a continuous helix, right? Even though these strands go elsewhere. All right, so we know we have base stacking. What else do we have? Well, we don't know where the blue helix is going to be relative to this yellow orange yet. So it could be parallel, so it could go somewhere like here, like I have pictured here. But it could also be twisted a little bit. So we could have it kind of going up like this or up like that, or maybe the other opposite way twisted, or maybe it's perfectly perpendicular, or maybe it's even anti-parallel, right? So we have a lot of different orientations. And this could actually matter if, for example, the orange and the blue have to bind a protein, right? So let's say there's a protein that perfectly fits here, right? If it was actually in this conformation, the protein couldn't come and bind the orange and the blue. So this type of kind of 3D arrangement of helices can actually become very important. So let's see what it is. And this is a 3D structure. It should spin a little bit so you can see it better. So we have that top base stacking. And then the blue is pretty much perpendicular, but slightly off perpendicular. So this is what they ended up solving. And I'm just going to briefly talk about kind of the forces that may be causing this. Now, we don't know for certain if it is, but one huge energetic thing to consider in RNA is that it's unfavorable for it to compact. And why is that? Well, here's just our RNA again, but I've made in very large red spheres negatively charged backbone. So RNA is a highly negatively charged molecule. You can see every single base has these negatively charged spheres. And we know negative repels negative, right? So this helix, if it was to come closer to this helix, that would be a repulsion and it'd want to move away because it would be negative charge next to another negative charge. So that makes it unfavorable to compact unless we have some sort of good interaction that makes them want to come closer together. So this is what we call tertiary interactions. It's often things like hydrogen bonds, you see a base pairing or those type of things. So in our structure, we actually do have one of these tertiary interactions. If we zoom in, so I've just highlighted, I think it's 6 bases in this case. So these are just GC base pairs and a helix in the orange helix, right? So what we're used to seeing, so they are base paired, so they're hydrogen bonded. So this is just like Slava highlighted the hydrogen bonds, same thing here in orange. But they're also hydrogen bonded to these adenines in this green loop. So this green loop is actually brought close to this orange helix because of these nice hydrogen bonds. So to summarize, we have repulsion of backbone. So that will kind of default is to have your backbone as far away as possible, except if you have some of these good interactions like base pairing, base stacking, other hydrogen bonds. So you can have base pairing between A's and C's, for example, sometimes. So those are what we call non-canonical. You can actually have triples. So you can have 3 bases all hydrogen bonding together. You can have adenines, like we just saw, interacting with a helix. And then you can also have things like metals coming into play. So these are all just good interactions that allow the RNA to compact and fold up despite the repulsion. So thinking about this, we can kind of, in a not precise way, but in a nice way to think about 3D structure, we can think of it as the addition of 3 things. So first, base pairing. Then we have base stacking. And finally, we have other tertiary interactions, or the lack thereof, which then the repulsion would take over. So if we build this up, we can see we don't want to represent it as kind of this nice 4-way junction. If we look at the 3D space, we actually have the yellow and the orange kind of making a continuous helix. Then we have the blue slightly off perpendicular. And then we have this green helix coming and interacting with these GC base pairs. So briefly, the method that was used for this is NMR. So the RNA is in water. It is limited to small RNA. So when you're looking at things that say they're solved by NMR, you shouldn't expect too large RNAs. And then one thing for practical purposes is it will pretty much always have multiple models in the PDB. And these are all consistent with experimental data, so they're all valid structures. So for example, when I opened this in Chimera, I would see all 10 models in this case. And this is what it would look like in Chimera X in this case. So I'd have a group of 10 different models, and you can see they're slightly different, but they're all valid models. So, you know, when I'm looking at it, I'll often just look at the first one, but all 10 are valid models. All right, briefly, let's go to case number 2, which is Pseudoknots, which I know there's a current challenge on, so I highly recommend people to go play around with Pseudoknots. So I'll just go through quickly the frameshift stimulation element from SARS-CoV-2, which is a great example of a pseudoknot. So its function is it actually causes the ribosome to slip sometimes. And this is what its secondary structure looks like. So this is what base pairing looks like. So we start with a green helix here, and then we have that blue loop there. Then we have a orange helix. We have a pink junction region, so here. And then we finish off this RNA. Now, if you notice through my coloring and your knowledge of base pairing, we can easily see that this loop can actually base pair with this region here. So it forms another helix. Now, when a loop forms another helix, that's when we call it a pseudoknot, because it's kind of knotted in a way, right? That loop is no longer free, and it's constrained to be near this other part of the RNA. So you can imagine, for someone that thinks about 3D space a lot, having an interaction like this, a pseudoknot, really constrains the shape that the RNA can make in 3D, because this loop now has to come near this far away part of the RNA. So these are great targets if we're interested in kind of nice compact RNAs. And the method that this structure was solved in, the 3D structure, which I haven't shown you yet for a reason, is cryo-EM. So that's what I work on. The RNA is also in water like NMR, but it's frozen water this time. Unlike NMR, it's actually easier with larger, less dynamic RNAs. And at least when we deposit the models, we'll also do the multi-model method. And for us, that's because we'll get volumes that are low resolution, or that means we see less detail. So we're uncertain on our models and want to make sure we fairly represent our uncertainty by depositing multiple models. So briefly, what is resolution? I just thought I'd show you some images and the scale. So the scale goes kind of in reverse numerical. So 10 angstroms is low resolution, 1 angstrom is high resolution. At something like 9 angstroms, you can see a helical shape. So you see here a nice helix. It's only when you get down to something like 4 angstroms that you can start seeing a backbone pattern. So at 4 angstroms, if you're looking at a structure that says it's 4 angstroms, you can be certain that they're putting, part of the RNA in the right position because they have the backbone. But notice they don't see a separation between bases. So their exact positioning of bases or what hydrogen bonds are happening in the bases may not be as accurate. It's really 3 angstroms that we start to see separation between bases. And then you can say, okay, they know where they're putting all of these bases. And then as we get higher, we can see cooler things like ions, like water. Like if you go to 1 angstrom, you can see individual atoms, which would be super cool. Yeah, so this is the shape. So this is the cryo-EM volume we see. So this is experimental. And then this is how we model it in. So we're just tracing the secondary structure now. We see a nice orange stem here, green stem here, and it forms this lambda shape. Now you notice because of that pseudoknot, so this blue loop had to bind to this blue region, we really get a nice compact form, right? So it's this nice little compact form that in fact, you could imagine if we're trying to drug SARS, we can have some holes here that maybe we can fit a drug because of its compact form. And I just want to note, don't forget, like in real biology, RNA is never just chilling alone. There's always other things around. So in this case, this RNA interacts with the ribosome, right? And we're lucky enough that the band group actually solved the structure on the ribosome as well. So adds an extra layer of complexity, but it's something that we should not try to forget. All right. So briefly, I've kind of talked about 2 techniques, NMR and cryo-EM. But where do most of our structures come from? So this is a breakdown of the database of all of our 3D structures. So on the left, I have RNA-only structures. And then the 3 main techniques, NMR, cryo-EM, and X-ray crystallography, which is still the most abundant technique that we get RNA-only structures. Now, if we look at RNA with proteins, so the ribosome is a great example of an RNA structure that has proteins. Cryo-EM becomes a more common technique. Very many of those are actually ribosome structures, but there's also other cool RNA protein complexes as well. So X-ray crystallography still, if you're looking at RNA structures, is one of the most common techniques you'll see. So what is it? The main difference I would say between NMR, cryo-EM, is that the RNA is in a crystal. So it's not floating around in water. It's in this compact form next to other RNA molecules. So to get it into a crystal, they often have to stabilize it to make it non-dynamic. So it's really kind of in a nice compact form. So this could be mutations. They could add some additives. And the practical thing to note is that when you download the PDB and open it, there may sometimes be multiple copies of the RNA in that model you open. So this is just a product of how they solve this structure. They may solve it as what we call a dimer. So that's 2 copies of the RNA instead of a single monomer. So just note that if it's not in a crystal and in water, it may not naturally be in a dimer form. This is really a product of crystallization and not necessarily would happen in water. Okay, so this is the PDB website. You saw it already briefly in Slava's presentation. We call it PDB. It's a different acronym, but yeah, that's that. And this is what a page will look like. So you'll have the unique ID up here, and then kind of the name of, in this case, the RNA. This is a ribo switch up here. And then some other things I just want to briefly point out. If you're interested in looking kind of what the experimental data looks like, you can often download it directly from files, but sometimes you'll have to come here and go to another database that has that experimental volume. The method is here, so you can know, is it crystallography, is it EM, is it NMR? The resolution is here, and this I think is an important factor, especially if you have multiple structures you could be looking at. You want to tend towards the one with higher resolution. Validation metrics, so blue is good, red is bad. This is percentile, right? So if it's 50 percentile, it's kind of average. So this tells us how good the RNA structure is, how physically realistic it is. And then the paper is always also cited at the bottom if you're curious to go back to learn some knowledge about the RNA that you're looking at. So again, just briefly to wrap up, some key thoughts. We can learn a lot more about RNA biology if you have 3D, but it's quite hard. We actually don't have that many structures right now. The 2D structure is often necessary to learn more about the 3D structure. So, we would love to keep improving on our base pairing predictions. The PDB is like this great source for 3D RNA structures. We currently don't know if natural RNAs have 3D folds. It's a problem with only I think it's 1,700 RNA structures total. We just don't know if many RNAs have a fold. And also due to the lack of structures, we also don't know if we've discovered all of the different shapes RNA can have. And the most important part is just, any thoughts or takeaways that, you guys have been inspired by this presentation. And science is a community effort. I think I don't have to tell a community like Eterna this, but also for us in academia, we have, a huge amount of people that, either directly work with us on our research or just give us advice or support. So here's just a listing of my present lab members in both of my labs, as well as past collaborators and other lab members. So I'm happy to take any questions, or honestly, I'd be happy with comments or ideas for Eterna in general, 3D Eterna in general. Oh, and I have not been reading the chat, so sorry if I missed anything.
No, that's too much. You can't.
Yeah.
While you're trying to present, that's too hard. So we did have one question about, is there a 3D modeling tool for over 500 NTs in particular? I guess so. The general question is there I know 3D modeling is not accurate. It's very early stages, which is kind of where we're all heading with this someday. But wondering if you have something that you like to use.
So specifically for longer, was that I hear?
Yeah, he's asking for over 500 NT.
Yeah, I mean, honestly, I don't work on anything long. So I've never tried to model something. I'd say probably 300 is the longest I've tried. So for me, I use Rosetta modeling to do smaller RNAs. Now, that process, as you get larger and larger, gets harder and harder and will take more computer power. So I don't think it's realistic to do something 500. So yeah, unfortunately, I can't really answer that question because I just don't have the experience with long RNAs.
So you said Rosetta, that's the open source Rosetta. Aren't there several different tools in Rosetta, or is there?
Yeah, so there's a couple. So the main one, I guess, to get a good starting point I use is called FAR2. So that's kind of a nice, you input the secondary structure and the sequence, and it will go and create a bunch of models and then rank them by energy. So you can look at your low energy or most stable 3D structures. For more like fine tune, if you really want to get like those hydrogen bonds perfect or that tertiary interaction perfect, we use something called stepwise, what is it? SWM. I think it's called stepwise in Rosetta documentation. And that is more time consuming computer wise, but will really get the details better than far too low. So those are the main two we use. There's actually in the past like 2 months been a lot of new papers, especially people trying deep learning algorithms on RNA. But the evaluation of those has not quite happened yet. It will after CASP is done, and we'll be able to see how they're actually performing on blind challenges. So that will be exciting, but I don't know how they are yet.
Well, I was wondering, it seemed yesterday Jennifer was saying that temperature had an effect on the 2D structure, and would it have an effect also on the 3D structure? So would you expect it to change when you freeze it, or even more when you put it in a crystal?
Yeah, that's a great question. And that's actually one of the reasons I started cryo-EM, is for that exact reason. So, you know, Jennifer was talking about, you know, ensembles of secondary structures, right? And you can imagine if the secondary structure is not just the low energy, if we have multiple secondary structures, we have even more possible tertiary structures, right? 3D structures. So that's certainly a concern. And that's actually why, so in the crystal, right, you're just stabilizing one form. So yes, it may be the lowest energy form, which still gives us information. But my goal maybe in 10 years is that we can have a true kind of ensemble view of 3D structures. Cryo-EM can potentially get that to you. So as a very top level, we image millions of different RNAs. And then so they're all perhaps in different conformations. One may be a little more up, one a little more down. And then if we use enough computer power, we can separate those out and get, you know, the up form, the down form, because we're not in a crystal. So that's kind of what I'm hoping cryo-EM will be able to enable. But currently we're just not there yet. So,
All right, thank you.
And I think Astronaut wanted to ask a question. If we can unmute him. And then I have a question.
Yes, I was wondering what you meant by we didn't know if nature folds in 3D. Yeah, I thought the 3D model was showing us how nature folds.
yeah, so yeah, I didn't really say that very clearly. So let me try again. So let's take the SARS-CoV-2 genome as an example, right? It's 30,000 bases long. Presumably some of those regions are what we call non-structured. So they're, you know, not really base paired that much. They're kind of just moving around, flopping around, look more like strings. Presumably some of the genome is highly structured, has 3D shape, and interacts with like either viral proteins, interacts with human proteins, has some function that the shape is important for. And the amount or the percentage of natural RNAs that actually have a shape that's important for its function versus ones that are kind of just flopping around and the shape doesn't matter, is currently kind of unknown. We really don't know, partially because we had to try to crystallize everything before to try to get it to see if it has a shape. So does that make a little more sense?
Yes.
Yeah, okay, cool.
Thank
Thanks.
you.
Yeah, so Rachel, cryo-EM, maybe you covered this a bit in your talk because I had to do something else while you were talking a bit, but so I think cryo-EM is new to DOS Lab, And that's going to be something that the DOS lab is going to be able to do going forward.
Yeah, I mean, that's the goal. So yeah, so right now I'm the main person in the DOS lab that can do the Crowley pipeline, let's call it, so get structures. But yeah, the goal is to have this as, you know, a major technique that complements both the DOS lab's computational work as well as their secondary structure work, to learn more about RNA structure.
That's really neat. Are you currently, I don't know how to say it, freezing? Is that right? Are you currently working on a structure for something right now, or are you, is the DOS lab still ramping up for it?
Yeah. So yeah, I still solve structures currently. So we still have some SARS-CoV-2 structures that hopefully we'll be getting out soon. I'm particularly interested in viral RNAs. So that's kind of where that goes. SARS-CoV-2, there's some in HIV, influenza. But a lot of current cryo-EM is really trying a lot because the technique is so new and, you know, it's pretty hard right now that, you know, we try 5, we get 1 of them to succeed. So that's kind of where we're at right now. But my focus is mainly viral RNAs. So.
And when you're looking at the SARS-CoV-2, are you doing cryo-EM on like a 300 nucleotide length, or is it shorter or longer? Or
Yeah. So I'd say 300 is kind of the maximum, mostly because at least when we look at computational predictions, the structural units, so what kind of folds in a nice compact form, is often closer to 150, or it really depends on the region, right? But and our lower limit, so we do have a lower limit. I think currently the lowest that's been solved is around 70 nucleotides, and I really should know this because I worked on that, but I can't remember the exact number. So that's kind of like the smallest we can go with cryo-EM right now. So that's kind of the range between 70 and 300-ish.
okay, that's really helpful to know. And then we go back to something we were talking about yesterday, and Reju said to ask you is, we're thinking about Eternal, and it came up, well, we're doing Pseudoknots in NewPack, and can we eventually get, I mean, someday hopefully we'll be able to do Pseudoknots in other modeling engines and maybe a different version of Eternal. I don't know. So Reju said there's a way to use base pair probability output in Eternal with Thresh-Knot pipeline.
Yeah.
Okay.
Yeah. So first of all, pseudoknot prediction is like, compared to normal base pairing prediction, is like years behind, in my opinion. Like it's a hard problem. We don't have great data to train all this stuff on. But so if you have the base pair probability matrix, you can essentially have heuristics, and Thresh-Knot is one of those. What Thresh-Knot does is it takes the mutual maximal base pairs. So it takes like, okay, these 2 bases like base pairing together, let's have them base pair. And in doing that, it can form pseudoknots as well as non-pseudoknotted structures. So yeah, if you guys are calculating base pair probability matrix, you could definitely use that Thresh-Knot to predict pseudoknots out. It would totally work. Whether they're correct, I don't know. But
What do you think, Jonathan? Can you talk to Rachel about that more later? Or
Possibly.
okay.
Definitely have to look into it.
Yeah.
All right. Well, we went 10 minutes long, and Eli put something in the chat, and maybe Rachel can look at that later and talk to him about it.
Yep.
Lorenzo Brani asked, how are non-canonical pairs taken into account for pseudoknots? I've been kind of looking at that in NewPack.
Strictly speaking, not any more than non-pseudoknotted structures, as far as I know.
yeah, I think as far as I know, that would probably be my answer too, Jonathan.
Thanks so much, Rachel. That was
of course.
I
Yes,
should
thank you very much.
just say first, that was really great. All 3 presenters this morning. Thank you so much, everyone.